process هذه مهمة spermatogenesis. And spermatogenesis starts with haploid spermatozoa. Okay, haploid spermatozoa, and this haploid spermatozoa will develop from germ cells, okay, and form a result that is called spermatid by a series of mitotic and meiotic division. Okay, إذن هذا الفيجر مهم. عندنا mitotic وmeiotic division. Okay, mitotic and meiotic division. البروسيس اسمه spermatogenesis يبدي بهابلويد spermatozoa. هابلويد spermatozoa يتكون من spermatogonia وينتهي بسpermatid. واضح؟ إذا النتيجة الأخيرة للspermatogenesis is spermatid. And the kind of the division is will undergo mitotic and meiotic division. Okay, so we will have 3 divisions in order to have a spermatid. Then we have another process which is called spermiogenesis. Okay, spermiogenesis راح يعطينا النتيجة الأخيرة اللي to have a mature sperm. Okay, mature sperm cells. So the final stage of spermatogenesis is called spermiogenesis. And the final result of spermiogenesis is a mature sperm cell, okay? Regarding the production of the eggs, we said before, the ovaries are the gamete-producing organs, all of you know it. And the maturation of the oocyte begins before birth. So it means that whenever the embryo is a female fetus, the eggs, or let's say the oocyte, will start to develop or mature before the birth, and it will continue after the birth, whenever the adulthood start. Okay, so oogenesis is the process of the differentiation of the oogonia, or let's say oocytes. Why? Because we say oogonia is the first step for the formation of the primary oocytes. Okay, so the oogenesis process whereby oogonia differentiate into a mature oocyte. Okay, so primordial germ cells. So we said before a term that is called germ cell means that a specialized cell. Okay, so as you see right here in this figure, we have also mitotic divisions. So we're going to have an oogonium, then this oogonium will differentiate into the primary oocyte in prophase. طلاب إذا أي شيء تكتبون على الشات أو raising hand أنا ما بدي أشوف، مجرد أشرح السكرين، فاللي عنده شيء خليه يحكي، لأنه ما أقدر أشوف. أوكي. So قلنا عندنا مصطلح ثاني primordial follicle. We have a primordial follicle term. And this is actually whenever an oocyte, in the simplest way, an oocyte, a primary oocyte, will surrounded by flat epithelial cell. Okay, flat epithelial cells. We call this structure as primordial follicle. هذه مهمة نجيبها 100%. Okay، بما إنه إحنا لازم الأسئلة تكون هواية. فأحدى من الأسئلة تكون primordial follicle is a primary oocyte surrounded by flat epithelial cells, as you see in this figure. أو ممكن نجيب هذا الفيجر. أسألكم, this structure or this cell is known as مثلا primordial follicle, or let's say primary oocyte, or ما أعرف يعني أعطيكم اختيارات. Okay, زين. على السريع هذا الفlat cells. They will develop and they will be cuboidal cells. They will surrounded by more than one layer of these cuboidal cells, and they will form a connective tissue. Okay, then we're gonna see this zone that will surround the oocyte. We call it zona pellucida. Okay, zona pellucida is important to know that it has many important functions or duty. Okay, So whenever the primary, or let's say the primordial follicle or the follicle was surrounded by the cuboidal follicular cells, we're gonna see the zona pellucida. Okay, the zona pellucida. So at puberty, these follicular cells that are surrounded by many connective tissue and cuboidal epithelial cells, they will begin to accumulate fluid. راح يجمعون fluid. بما انه تشوفوها احنا عندنا primary oocyte surrounded by cuboidal cells. Okay, and we're gonna have a zona pellucida. من يوصل البوبرتي تايم راح يصير راح يبدي الفوليكولار سيلز يجمع لنا سوائل. Okay, begins to accumulate fluid where in a space that's called antrum. احنا راح نسمي هذا السبيس follicular antrum. Okay, whenever this antrum starts to grow up and let's say to be larger, it will be filled with some fluid, and this will give us a theca interna and theca externa with a tumulus oophorus. Look at here. We're gonna have a large antrum. بس أريدكم تعرفون راح يصير عندنا large antrum pushing the oocyte to a side. Okay? راح يصير swollen. ليش؟ لأنه قد عملية احنا نحاول إنه نحرر هذا الoocyte or let's say this follicle. نحاول نحرره. راح يصير عملية تجمع السوائل in order to push the oocyte to a side and help the oocyte to release. Okay. Besides this, there will be some contraction in the ovary, and this will lead to release an ovum from ovary. Okay, and we call this process as ovulation. إذا بمساعدة التقلصات الموجودة بالأوفري plus راح يصير عندنا some accumulation of the fluid in in the follicle that we call it mature Graafian follicle. This will lead to have rupturing and let's say like having an explosion and lead to release the ovum from ovulation. When this process happens, and ممكن أسأل when ovulation start to occur, you can see at puberty. Okay, how by the pushing of two process by filling the antrum with fluid plus the contraction of the ovaries. So we will say again, ovulation is release of the ovum from ovary. Simply تقدرون نجي تعرفوها. تمام. After ovulation process راح يكون عندنا طبعا ovulation قلنا بعد ما يتحرر الأوفم راح تبقى عندنا خلينا نقول traces of the follicle يعني آثار plus cuboidal epithelial cells that's surrounding the oocyte, let's say before. So this will form a corpus luteum structure. Then the corpus luteum or lutein cells عندها وظيفة اللي هي secreting estrogen and progesterone. Okay. إذن هذا السلايد هو إنه تعرفون corpus luteum راح يتكون after ovulation. وظيفته إنه secreting estrogen and progesterone, which both are important female sex hormone. So together with progesterone and estrogen راح يساعدون اليوتراين اللي راح enter a stage. واحدة من هاي الستيجات راح نسميها secretory stage واللي يسوي preparation for implantation of the embryo in the case if there is pregnancy case. Okay, so ovarian cycle تعرف ovarian cycle لأن هذه كلها فد عملية فد cycle يعني نسميها ovarian cycle. This includes ovaries preparation, or let's say the ovary will prepare and release an ovum in a series of events. Okay. هذا بالنسبة للمحاضرة الأولى. عندكم أي سؤال؟ ولا أكو سؤال؟ ما أكو سؤال أنا راح أسأل. اوكي نكمل. المحاضرة الثانية اللي هي fertilization process. Okay. The fertilization process, it has a simple introduction. A definition which can be said is a male and female gamete fulfills to produce zygote. راح نقول an ovum, a mature or immature, let's say sperm, they will fuse to form a zygote. Zygote, one أقول embryo. Tamam? Zygote, because the first production of the fertilization is a zygote. Okay. The location for this, we said before, it's ampullary region of the fallopian tube. We need 2 steps in order to have an effective fertilization process, or let's say to have a spermatozoa that can fertilize an oocyte. These 2 steps are capacitation and acrosome reaction. Capacitation and acrosome reaction. Capacitation is conditioning period, okay, where in the female reproductive tract. The second one is the acrosome reaction is releasing an enzyme that needed to enter the zona pellucida. So as you see, a zona pellucida work like a membrane surrounding the oocyte, which prevent any other sperms to enter to the oocyte. Okay, so these 2 steps are important, which is capacitation and acrosome reaction. زين نجي على ايش؟ 3 phases of fertilization. الأولى هي penetration of corona radiata, then penetration of zona pellucida. The last one is fusion of the oocytes and sperm cell membrane. Okay, هذه مهمة جدا. 3 phases عندنا: penetration of corona radiata, penetration of zona pellucida, and the last one is the fusion. Okay, and the phases are explained in detail. So خلينا نشوف اللي ممكن أسألكم إياها. مثلا هنا نقدر نقول النقطة الأولى regarding the corona radiata. The capacitated sperm pass freely through corona cells. Incapacitated, or أقدر أقول non-capacitated sperms can pass freely. طبعا answer is true or false. Yes, who can answer?
False, Dr.
Yes, good, thank you. Penetration of zona pellucida. طبعا zona pellucida هي عبارة عن glycoprotein shell. Okay, يعني glycogen زائدا بروتين. Okay. شلون هي عادة تحيط الإيج نفسه facilitate and maintain sperm binding. Okay, facilitate. zona pellucida شلون تقدر تمنع إنه ما حصل حالة بوليسبيرمي؟ هذه شارحيها قبل، تذكروا أعتقد. الزونا pellucida تغير الستراكشر والكومبوزيشن وتمنع حالة بوليسبيرمي. أوكي، تغير بنفسها حتى لا يقدر يتعرف عليها. زين، النقطة الثالثة اللي هي fusion of the oocyte and sperm cell membrane. طبعا بهالنقطة إنه راح يصير عندنا second meiotic division in the oocyte. ليش؟ لأنه بالprimary oocyte, whenever it's released, it was stopped in the first meiotic division. Okay, so شو راح يصير عندنا second meiotic division وراح يصير assumption وراح يكمل divisions in order to form the zygote. Okay, so the oocyte finished its second meiotic division. It was stopped in the second meiotic division, not the first, and will prepare itself in order to form the embryo. Okay, so the main result for fertilization, يعني أكيد the process يحتاج a result. After the fusion of these 2 main, the sperm and the ovum, we will have a result that can be summarized in 3 points, which is reformation of the diploid number of chromosome. راح يصير عندنا diploid number of the chromosome. Why? Because the two they fused and formed diploid chromosome. The sex of the new individual will be determined. والنقطة الأخيرة cleavage راح يجي. طبعا هذه وحدة ورا ثانية سريعة جدا، حتى الديوريشن ما نقدر إنه يعني نعطيها في وقت محدد يعني. اوكي. فالكليفج وقتها يدري الكليفج اللي هي series of divisions. اوكي. series of mitotic divisions، إنه الخلايا تبدي بشكل سريع تنقسم. اوكي. وين طبعا هي داخل الزيجوت، وتعطينا طبعا results of increasing in cells، وتكون يعني بطريقتين راح تصير انه holoblastic يكون asymmetrical cleavage. Holoblastic cleavage and asymmetrical cleavage. And holoblastic means that they will divide through their cytoplasm. And asymmetrical means that unequal in size cells. Okay, so the process of cleavage eventually will form. يعني طبعا هذا process of cleavage راح يستمر، بالنهاية راح يكون لنا structure راح نسميه blastula. اوكي. والخلايا اللي داخل هذه البلاستولا راح نقوم نسميها تسمية ثانية، نسميها بلاستومير. واضح؟ زين. البلاستومير راح تتجمع whenever this blastomere will be like a cluster of 16 to 32 blastomere cells. We call this structure as morula because it will look like a morula shape. Okay. Then this morula will have some differentiation. شوية حصل بيها differentiation بسيطة اللي هو المجموعة الداخلية. The inner cells we call it inner cell mass. Okay. And the outer cells we call it outer cell mass. Look, mass. Why? Because we have a mass of cells. Okay. Then we will have the structure that is called blastocyte or blastocyst formation. Okay. So the blastocyst or blastocyte came from morula. Whenever they differentiate, they will differentiate more and more in order to have an embryoblast and what? Trophoblast. So embryoblast will form the future embryo, or let's say the future fetus, and this trophoblast, which is from outside, will form the placenta. Okay, it will form the placenta, or let's say future placenta. واضحة؟ ممكن أجيب لكم figure هذه أو هذه، ممكن أي وحدة موجودة داخل المحاضرة. These figures showing you how the first cleavage, and then the second one, and then you can see the third one is having 4 cell stage, then early morula, advanced morula. راح يصير عندنا blastocyst. Then this blastocyst will be implanted راح تنغرس داخل الرحم. Okay. Any question regarding this lecture? Yes or no?
عفوا دكتورة، قلت راح اجيب سلايدات بس ما فهمت يعني شلون راح تجيبيها.
يعني هذه الصور راح اسالها مثلا هذه. اوكي. اختار اي شيء من عندها اسالكم عليها. ممكن امسح الباقي وابقي واحده مثلا. يعني الاستاذ من جايبته يعني شيء اجيب بصوره عامه وليس مو بشكل دقيق. واضح؟
بلى دكتور.
lecture number 3 اللي هو implantation. راح اشرحها على الوورد اللي عندكم. هذه يعني بشكل مختصره احسن من اللي خلينا نقول اللي عندي مفصله. خلينا نقول implantation. طبعا تعريفه مهم. شنو هو الامبلانتيشن؟ قلنا قبل شوي قلنا انه we said before in the second lecture that the blastocyst, which is the final structure before implantation, this blastocyst will implant, in the uterus. So the stage of the pregnancy at which the embryo adhere to the wall of the uterus is called implantation process. Okay, هذه كلش مهمة. So by the adhesion that the embryo will receive. ليش نحتاج implanting؟ ليش ما هو طافي داخل الرحم من دون أي اتصال؟ ليش؟ Because we need a supplement, isn't it? Like embryo receive an oxygen and nutrients from where? From the mother to be able to grow. هاي سهلة. وأكبر لكم السلايد. Okay. So this is regarding the second week of development. شفتوا انتوا the first week was fertilization, increasing in the cells number, having a differentiation to have a blastocyst, which is consisting of embryoblast and trophoblast. In the second week of development, we will have a differentiation in these two layers, which is the embryoblast and trophoblast. So regarding the trophoblast, I mean the day 8, the differentiation that happened the day 8, this trophoblast will divide into cytotrophoblast and syncytial trophoblast. Okay, cytotrophoblast and syncytial trophoblast هي نقول التمايز اللي حدث من trophoblast. Okay, so cytotrophoblast will be the inner layer and syncytial trophoblast will be the outer layer, let's say, or the multinucleated layer of the trophoblast اللي هذا راح يكون placenta بالمستقبل. So inner cell mass ايش راح يكون or what will have as a differentiation? They will have the epiblast and hypoblast. Okay, epiblast and hypoblast together they will form a bilaminar germ disc. Besides, we will have another event which is the epiblast cells. شوفوا هذا epiblast cell اللي هو adjacent to the cytotrophoblast. Okay, they will form a new layer of cells. We call it amniotic cells or amnioblast. Okay, they will form a cavity that is called amniotic cavity. Okay, amniotic cavity. So we can say that the origin of the amnioblast is epiblastic cells. So the small cavity, I mean this cavity is also called amniotic cavity. Also we can say that the origin of the amniotic cavity is epiblastic cells. Then نجي day 9 and 10 عندنا some events which is the most important is formation of exocelomic membrane, formation of exocelomic cavity, formation of the lacunae, plus we can say this lacunae stage is most important right here. كلش مهمة lacunae stage. Why? Because why it's important من يقدر يتذكر. Yes, who can tell me? على فكرة ما مطلع حضوركم. اللي يطلع يدخل يسوي هيك انتوا الخسرانين. Yes, who can tell me what was lacuna? In the simplest way. ما حد يتذكرها؟
دكتورة ممكن بس تعيدين السؤال لأن الصوت يمي ذا يقطع ما راح يكون من وين الدكتورة.
ما راح أعيد السؤال راح أنطي الجواب. Okay, so the lacuna stage is the first step in order to have a blood circulation between the mother and the embryo. The formation of the lacuna that will happen in the trophoblast. The trophoblast is the layer that will form the placenta in the future. Okay, the lacuna will fuse. These large lacunas will form. This cavities will form a large lacuna. نسميها trophoblastic lacunae or lacunar stage. They will be the first step in order to have the blood circulation process between the mother and the embryo. Okay, day 11 and 12 we will have the continuing of the blood circulation process. راح نسميها uteroplacental circulation. So after the formation of the lacuna in the trophoblast, we will see that the formation of the sinusoids will continue. Sinusoids is a penetration of to the deeper stroma and eroding the endothelial lining tissue, okay, and having the capillaries inside this lacuna, okay. So congested and dilated capillaries that will let's say extrude the lacuna will known as sinusoids. Then the uteroplacental circulation will begin, okay. يعني blood circulation will start between the mother and this fetus. So the most important question right here I always ask, the uteroplacental circulation begins in the first week of embryonic development. Is it true or false?
False.
False. Good. So the formation of extra-embryonic mesoderm, we will have some embryonic mesoderm, like somatic and splanchnic mesoderm right here, will surround, look, it's an extra-embryonic mesoderm. So it will surround the embryonic structure, the early embryonic structure, and have some duties in order to form some structures in the future. Okay? Then we will have formation of extra-embryonic cavities that can be seen right here. Look, extra-embryonic somatic mesoderm and extra-embryonic splanchnic mesoderm with the cavities right here. Okay? إذا تشوفون الستراكشر هو يوم ورا يوم يتمايز حتى يكون لنا الامبريو نفسه، أو تشوفون البلاسنتا شلون يحاول يكون نفسه حتى خلينا نقول حتى هذا الجنين يتشبث بالحياة وخلينا نقول يجي له دم أو يجي له غذاء من الأم. أوكي؟ عن طريق شنو؟ عن طريق طبعا increasing in cells number and specification differentiation of these cells. واضح؟ نرجع على day 13 اللي هي آخر وحدة. هي formation of primary villi. شوفوا primary villi, formation of secondary yolk sac, formation of exocoelomic cyst, formation of connecting stalk، كلها حتى في سبيل انه يربط الجنين بالرحم، حتى انه يجمع الغذاء وينطيها للجنين حتى يصير عنده خلينا نقول نمو development. اوكي، اذا primary villi حدث يحدث بالسيتوتروفوبلاست. اوكي، السكندري يولك ساك نفس الشيء يحدث عند hypoblast cells، باعوا hypoblast cells يعني مو بالمشيمة، اوكي، اللي هي بالامبريوبلاست. ليش؟ Because this yolk sac will help in the growing of or the development of the embryo. Then we'll have formation of the exocoelomic cyst اللي هي اللي هي large portion of exocoelomic cavity، راح تجي من exocoelomic cavity then pinched off، يعني كأنه تتخصر من عندها وتكون exocoelomic cyst اللي هي هم نفس الشيء راح يصير بيها differentiation، وحالة من حالات إنه يساعد ال development of the embryo in this stage in the second week of embryonic development. النقطة الأخيرة اللي هو umbilical cord راح يبدي يتكون. شلون راح يبدي يتكون؟ عن طريق تكون connecting stalk. So the connecting stalk will form in the second week of the embryonic development, and this leads to form the future umbilical cord. شنو ممكن أسألكم من هاي النقاط؟ مثلا origin ممكن أسأل، أو مثلا أسأل how primary villi formed by proliferation process of the cytotrophoblast cells. مثلا ممكن أسأل secondary oxide. What's the origin of the primary villi? الأنسر هو cytotrophoblast. اوكي؟ زين، اللي هي جاية من ايش؟ جاية من trophoblast. النقطة الثانية هي formation of the secondary yolk sac. So what's the origin of the secondary yolk sac? You can answer is hypoblast cells. اوكي، لأن هو يقول لك hypoblast cells proliferation. النقطة مثلا اللي نقول exocoelomic cyst came or pinched off from exocoelomic cavity. Then the connecting stalk will form in the مثلا in the end of the second week of embryonic development. True or false? It's true. Umbilical cord formed from connecting stalk. True or false? True. Regarding the figures, الفيجر اللي موجودة هاي مثلا شوفوا الأسبوع الثاني أكثر شيء أهتم بيها إنه لازم تعرفون إنه uteroplacental circulation راح يتكون، تمام، ويصير عندنا من implantation process. فمثلا ممكن أسألكم هاي differentiations أو differentiations اللي يحدث بهذا الأسبوع هو مهم، إنه هو مثلا التروفوبلاست راح يصير سايتوتروفوبلاست، سينسيشيال تروفوبلاست، امبريوبلاست راح يكون ابيبلاست، هايبوبلاست، بايلامينر جيرم ديسك هو يبين في الأسبوع الثاني. regarding this I mean هذه الصورة، هذا الفيجر اللي تشوفوه اللي مهم من عنده هو اللاكينا، اوكي، اللاكينا وساينوسويدز، اوكي، اللي هي maternal sinusoids. هذه تروفوبلاستيك لاكينا maternal sinusoids. واضح؟ ولا واضحة؟ شوفوا آخر فيجر راح يشوف لكم هنا هاي النقطة connecting stalk، هذه مهمة أسألها، تمام؟ maternal sinusoid connecting stalk تروفوبلاستيك لاكينا، هاي ثلاثة كلش أأكد عليها. واضح؟
واضح دكتور.
عندكم سؤال طلاب؟ راح أنزل اللينك سجلوا حضوركم، أفتحها 5 دقائق طلاب. خلوني بس أسجل سكريني لأنه كلش مزعج.